Good afternoon, everyone. Thank you for being with us and welcome back to our research and winemaking webinar series. So we hope today you will enjoy that webinar and you will learn new skills to develop new knowledge to be used in your vineyard and winery. I'm Dr. Aude Watrelot. I'm an assistant professor and extension specialist of enology at Iowa State University. And I'm co-hosting this webinar with my friend and colleague, Drew Horton, who is a research scientist, specialist of enology at the University of Minnesota's Grape Breeding and Enology Project. So before starting the webinar today, just few instructions. The webinar is being recorded. So if you want to watch the previous recordings, feel free to go on YouTube and you will be able to see the ISU UMN Research and Winemaking playlist where you can see everything. If you have any questions during the entire webinar, feel free to use the chat box that you can find at the bottom of your screen to ask any questions. And then at the end of the webinar, we're going to share a link to a survey. So if you can take between 2 to 5 minutes max to complete that survey, that would be wonderful because that's going to help us just to improve the webinars for the next time. So today we will have two presentations. So we're going to switch between Dr. Richard Descenzo and Dr. Misha Kwasniewski. So we're going to start by Dr. Misha Kwasniewski from PennState University, who is going to present the impact of yeast strains on acidity of juice and wines, especially on non-Vitis vinifera grape varieties, for about 25 minutes. After that, we're going to have Dr. Richard Descenzo from ETS Laboratories, who is going to talk about the importance of wine microbiology and yeast and bacteria that are used in the industry for acidity management for about 30 minutes, so 25, 30 minutes. After those two presentations, we're going to have a Q&A, so question and answers, and a panel discussion with grape and wine industry members. So John Taylor from Granville Wine Consulting in Minnesota, and Kent Foulker, who is working at Cedar Ridge Winery and Distillery in Iowa. So let's start by the presentation of Misha.
And I'll introduce Dr. Misha Kwasniewski is the Associate Research Professor of Fermented Beverages at Penn State University. Before taking his position at Penn State, he worked at the University of Missouri, where low tannin and high pH, high total acidity wines were among the chronic challenges and problems faced by producers there. Misha's research focuses on understanding and remedying problems faced by wineries, distilleries, and breweries through the lens of analytical chemistry. He's also worked extensively to elucidate the relationship between growing conditions and flavors in fermented beverages. Very important research. Misha, thanks for being here. The floor is yours.
Great. Thanks, Drew. And if I'm doing this right, I think my first slide should now be up.
Looks good.
All right. Awesome. Well, thank you both and all of you for having me talk today. I'm going to start off with a bit of a rehash and a reminder of what's going on in the vineyard, because ultimately, anytime that we're talking about what's happening during fermentations and what's happening with high acid wines, it all dates back to that we had to have the precursors for what chemicals we're working with later produced in the vineyard. So generally, all of the acids that we're dealing with at the end of the season are produced fairly early on. Tartaric acid really just in those first couple of weeks after berry set, all the tartaric acid you're going to have for the rest of your winemaking is being produced then. Likewise, malic acid is also being produced fairly early on. And if we look at this sort of general view of what's going on in wine ripening, we see that tartaric increases until like malic takes over, and then right around veraison we get this switch where we go from a increase in malic to malic starts to degrade, and then sugars are accumulating. One of the interesting things that I ran into in my time in Missouri, and I'm sure many of you are dealing with it in Iowa, is that ideal Vitis vinifera model that works in California and works in most of Europe doesn't quite track with all the cultivars that we're working with. And so what I've got right here is some data that I pulled from one of my former grad students' theses, where on the left we have sugar content of Norton being grown in Missouri on the left, on the left top, and then Cab Sauv on the bottom. And we see that basically is following the trend that we would all expect to be seeing, that just as time goes on, as we allow more and more time for the fruit to be on the vine, we're seeing an increase in sugar content. Where it gets a little bit wonky, though, is that some of these cultivars that have some Native American heredity continue to increase in malic acid long after veraison. So we're still seeing on the top, we've again got that Norton. We're seeing what's happening with malic acid after veraison. And we see that that continues to go up for quite a while. And then it never really degrades at the same rate that we see with the Cab Sauv. Even within the exact same environment, we're seeing huge differences in the possible fruit chemistry that's coming to you at your winery door. So all of this ends up mattering because we have different acids within the wine, some of which have different organoleptic properties, some of them have different levels of stability, and we need to decide what sort of trade-offs we're going to make if we're going to adjust or try to digest some of those microbes. As a reminder, pH and TA don't really, while they often relate to each other, they don't necessarily correlate beautifully, because you can have a high pH and very high titratable acidic wine, or vice versa, depending on what's going on with buffering. And that different acids have sort of different inherent chemistry, with some acids having the ability to donate one or some two, like tartrate, one or two protons, like tartaric acid, and then some, like citric acid, actually able to donate 3 protons. So for 1 molecule of citric acid, you're donating potentially 3 times as much as 1 molecule of acetic acid. So just to sort of see where that pans out, looking at this again, we've got malic acid, the different pHs where they dissociate, tartaric acid, which has the ability to donate the same amount of protons, but slightly different points on the pH scale. And then finally, lactic acid, which is often, when we're talking about acid degradation or conversion, our final product. And we can see why that's beneficial, because if we go from malic to lactic, we've gone from donating 2 protons to donating 1 proton. So just again, is a little bit of a reminder of what's going on with these different acids, that in general, with tartaric acid, we expect to see about 2 to 6 or 7 grams per liter. Malic acid, probably some of you have had to harvest at over 10 grams per liter of malic acid, but we're hoping to get that a combined tartaric and malic, that it's at least coming in a little bit under 15 grams per liter. Lactic acid will never be a major acid in the fruit, because it's not something that's produced by grapes. So any lactic acid you have is going to be the byproduct of further microbial fermentation. This could and does occasionally happen, that you'll see a small amount just from rots and things that will happen on the fruit, but that's not still something that's happening from grape biology. Citric acid, which we come in contact with the most in, say, oranges and lemons and whatnot, also is interesting from a hybrid production perspective, because a lot of the hybrids tend to be much higher in endogenous citric acid, so the amount of citric acid in the fruit, than other cultivars. So we're all of us winemakers or grape growers, I'm sure, at least familiar with the basic idea of traditional fermentation is that sugar goes out into yeast and ethanol comes out. And when we're dealing with any sort of microbial fermentation, something has to go in for the yeast or bacteria or other microbe to derive energy, and something has to be excreted from that energy, that we're never able to, create or lose energy without something else happening. So when we start looking at alternatives for how either different yeast strains or utilizing different pathways, keep in mind something always has to be digested, and there's always going to be some product. Sometimes that's something that is good from a taste perspective. Sometimes that's something that's really sort of nasty and not enjoyable. When we look at more like, and this is just still a little bit of an oversimplified view of everything that's going on with general Saccharomyces cerevisiae yeast metabolism, we see, again, sugar can go in, however, also amino acids, and there's a number of other ways in which yeast can derive the building blocks for producing other products, and then a lot of things other than just alcohol come out. And we know this to be true because when we ferment wine, it's not just alcohol and water grape juice. We get all sorts of nice esters and other higher alcohols and aldehydes, and also things like hydrogen sulfide and acetaldehyde and acetic acid. So some good, some bad. If we want to select for yeast that produce lower acetic acid, for instance, then we're selecting for other pathways to be preferentially used for these products. One other just fact that I want you to keep in mind of sort of the basic chemistry of what we're talking about as we're dealing with acids is that not tartaric acid does not act the same at all pHs, partially due to that pKa instance. And so tartaric acid at a lower pH is more likely, if it precipitates, to cause the pH to go even lower, whereas at a higher pH, it's probably going to pull that pH higher. So as we have interventions with certain yeasts, there's ways in which that can be either beneficially used or not. Sorry, this is a little bit caught up, but I think one of the interesting non-Saccharomyces yeast that's now becoming, there's options that are commercially available, uses the fact that it has a pathway that rather than sugar going in and ethanol coming out, it's able to digest malic acid into ethanol. So this has a number of potential interesting implications, one of which being it is directly degrading that ethanol. And unlike doing a traditional malolactic fermentation, you're not converting just one with two protons into an acid with one proton. You're causing the malate altogether to be digested and turn into alcohol. which will then have implications on potassium bitartrate ability, mouthfeel, and a number of other factors that might sort of round out that wine. The flip side that we should all be thinking about anytime you start to digest something new is what is that potentially going to do? If you have a wine that came in with 7 grams per liter of malate and you use Saccharomyces and you're using a strain that's able to fully digest that malate, you now have the potential for a wine that amazingly actually is going to be a bit on the flabbier side and also may have taken on some unwanted character that you don't traditionally want with your winemaking. This genus is one that's been sort of lauded as well as some others for, potential benefits of wild fermentations or natural winemaking that you're using a whole host of selection of different genus rather than just Saccharomyces. But one of the problems that comes up over and over again is that in the interest of trying for something new, often you get some unwanted volatiles and other character. And specifically with Saccharomyces, many of the wild types are very high acetic acid producers. The cool thing, I think, and one of the exciting things that whether it's this genus or some of the others that are becoming available is just like not any Saccharomyces cerevisiae is good for making great wine, not just any one of these strains is necessarily going to give you the qualities that you want. And so as yeast producers and researchers start combing all of the diversity that there is out in the world for these different strains, they're finding ones that have less of the deleterious qualities and cause some of the more beneficial aromas and whatnot to happen. So with that, I'm going to switch over to talking just briefly about the potential and some of the challenges of mitigating wine acidity through bacteria. And so most prevalent malolactic bacteria that's being used is Enococcus eini. It has some good points and some bad points, and generally, many of the early strains were not terribly resistant to sulfur dioxide. They tended to only ferment very well at fairly high pHs. However, they did better than many of their counterparts. And if we look at roughly what is happening during the different phases of metabolism of Enococcus eini, we see it in its first phase of growth preferentially digests any sugars that are left behind. So if we're dealing with a wine that is already bone dry and we're doing a sequential, meaning primary happens, finishes, and then we put in Enococcus eini, it's probably not going to be able to do much of this initial catabolism. Now, if we're doing a simultaneous digestion, Enococcus eini is probably going to be conducting more of the fermentation of sugars than it would later on. As carbon sources start to get used up, Enococcus eini preferentially uses malic acid and then citric acid. 2 points that I'd like to make here that are really critical when thinking about hybrid wines and very much high malate hybrid wines is all of that malate has to go somewhere. And if you're dealing with a hybrid wine, especially things like Chambourcin, I've seen just astronomically high levels of citric acid. That citric acid also needs to create a byproduct. In the case of malic acid, you're getting lactic acid, but you're also going to get a lot of compounds like diacetyl. With citric acid, it's a huge amount of diacetyl. So you can create something that is very, very out of balance and highly buttery if you allow that malic acid fermentation to go right through all of the phases. Additionally, since you're putting these bacteria into a fairly challenging environment, if you're asking them to do a lot of fermentation, you may need to worry about some of the nutrition requirements in a way that you wouldn't have if you're going off of the recommendations of common vinifera fermentations. One sort of glimpse of other possibilities that are on the horizon. In general, I would say, just like Saccharomyces, that we're sort of in our infancy of selecting new varieties and figuring out how best to use them for primary fermentations. We're in this similar place when it comes to bacteria to use during fermentations. And so Lactobacillus plantarum is one option that's been out there and has been found in wine actually for a very long time for doing ML conversion. However, lots of the varieties that have been isolated tend to peter off at lower alcohol, so they don't really work at all sequentially, and they have nowhere near the pH resilience that Enococcus eni strains that are currently in use have been. However, new species are being found, and they're also being found to have some pretty cool new activity, like this study that found that there are lots of Lactobacillus plantarum that have glycosidase ability. So that ability to cleave off those aroma compounds that otherwise would not be volatile. Many of the really important flavor compounds that are derived from the fruit otherwise might not be getting out in your wine and presented if you don't have some glycosidase activity throughout the fermentation. And so I think paired with some of the other alternatives and working it into an entire system, there are some possibilities for Lactobacillus plantarum to really make new wines out of many of the cultivars that we're already growing. The last point that I want to get into is, as we're thinking about how to manage acidity and sort of adjust wines and make our best wine out of the fruit that we have, we need to be thinking about how that balances with many of the other components that are going on. So microbial management of acids does not just come from acid degradation. It comes from how we're farming. It comes from potential interventions. It comes from blending. It also comes from how we've wholly balanced the wine. Additionally, there are all sorts of chemical aspects that relate to the microbiology. If you even do something like chapitalize, you now are changing the entire system for what sort of solubility you're likely to be seeing later on in fermentation. From a flavor perspective, I think it was really well sort of put in something that I think of frequently anytime that I think of in when I'm talking about balance, is this figure from Emile Pinot that frequently we think of this continuum of acidity and sugar, right? So if we have lemonade, we add enough sugar, eventually it becomes imbalanced. However, with wines, we have other factors that are actually at play. And weirdly, alcohol is a nice counterpoint to acidity. If you've ever had some of the non-alcoholic or even more frightening, some of the de-alcoholized wines, you'll notice that there's this intense acidity that comes through. And that's because that alcohol is actually sort of deadening the perception of acidity. Likewise, if you have absolutely no tannin to a red wine, which is something that we fight with quite a bit in cooler climates, and certainly run into this in Iowa and elsewhere, is that if we take that tannin out of the mix, even if we're not trying to make a highly tannic wine, the balance is all sort of thrown askew. So as you're thinking of how to build a wine, don't just think of how do I increase or decrease one of these components. Think of how if you pull that equilibrium somewhere else, how you may be able to make a balanced wine out of something that otherwise wouldn't quite have worked. So with that, main conclusions that I'd like to have you take away with, there's new selections of microbes are becoming available all the time. Keep an eye out, keep excited. However, also keep cautious. As you get into using anything that isn't your tried and true, remember those early days of winemaking when you were terrified that you weren't going to have a complete fermentation. As you work with new microbes, that's a challenge. That's a reality that you can have stuck fermentations, can have weird spoilage microbes getting in. And at the end of the day, while we always want to make the best wine we can, we also want to make wine and not vinegar by the end. There's also always going to be a trade-off for products that you're going to be producing from any metabolites consumed. And that many of these microbes will have different tolerances and requirements than those that you've been used to. And then finally, think holistically, that decisions you make early on, whether it's in the vineyard or early on during fermentation, need to be in balance with your ultimate outcomes, and thinking about how that might impact calcium tartrate stability or potassium tartrate stability or other elements of microbial stability, and what you're willing to give up or trade at one point to be safe at another point during your winemaking. So with that, thank you. And I believe we're going to go right into the next talk. Or
Well, I'm going to thank you for that talk. And
Thanks,
I had no idea I still had so much to learn about winemaking. That's very interesting considerations to think about for the future of winemaking, timing of secondary fermentation, all that stuff. I look forward to our Q&A and our discussion afterwards. So now I'm going to introduce Dr. Richard Descenzo from ETS Labs. Dr. Richard Descenzo is the microbiology group leader and co-general manager for ETS Labs in California. ETS Labs provides a range of chemistry and microbiology analyses to help clients in the winemaking process. In addition to providing microbiology support for the wine industry, Rich focuses on helping clients, including me, with a wide range of other winemaking problems. Rich received his master's degree in plant pathology and a PhD in plant biology at the University of New Hampshire, and conducted his postdoctoral studies on disease resistance genes in barley with the USDA ARS at Iowa State University. He spent 10 years conducting research on grape genomics, development of molecular diagnostics, and fermentation monitoring in the wine industry prior to joining the ETS Labs team. Rich, the floor is yours.
All right, let's see if I do this correctly. There we go. All right, well, let's see. I guess it's good afternoon for you folks, and it's actually good afternoon for me. I gave a Zoom seminar for UC Davis this morning from 9:30 to 11, so it's already noontime or afternoon. So welcome everyone. Misha, that was great. You laid out a lot of groundwork, so I don't have to explain some things nearly as much, so I appreciate that. when I was asked to talk in this seminar, I was primarily asked to talk about the microbial manipulation of juice and wine acidity. And so sort of to go on from the previous discussion, that's what we'll do here today. And once again, thanks for having me. It's always fun. I've got to figure out how to advance. There we go. There we go. Okay. So I always start off in every talk when it has to do with microbiology and chemistry, just to tell people that, you know, juice chemistry, we all know, has a profound influence on wine chemistry. Juice microbiology impacts juice chemistry and also wine microbiology. So it's all tangled together. And so what comes in on the juice doesn't stay in the juice. So it's really important that whenever we're looking at any topic, especially something like acidity or acidity management, you have to start with the juice. Okay. And one of the things, this is just purposes of this slide is just to demonstrate that juice chemistry can vary greatly from vintage to vintage. So every year I crunch the numbers for all the varietals and make these scatter plots and then, you know, give seminars on what we saw the previous harvest. And so these are some that we looked at. So this is 6 vintages, 2018, I think, through 2023. I left out 2000. Oh, no, I guess I didn't include 2020. That was the year of the fires. That was an odd one. But just to show you, you know, overall, juice chemistry profoundly influences wine chemistry. And if you can see here, this dark blue and yellow line, so that was essentially 2023 and 2018. Okay, and those were cool vintages. And you can see by the scatter plot, it was a much later harvest. And those cool vintages had lower pHs. One of the things we also see associated with these cooler vintages, lower pHs, are higher overall malics and lower potassiums. So once again, every year the juice coming in can be different, and most likely it will be different. So it's important to know what's going on when you bring the fruit in. I wanted to talk a little bit about organic acid profiles. This is an analysis we run on both juice and on wine, simply because some of these topics, some of these, these acids we'll talk about as we go forward in the seminar here. And so in juice, if you look and compare versus wine, we generally see average citrics are higher. You know, gluconics can be higher. We see, it doesn't seem like a big difference in glucuronic. We always see higher lactics, lower malics, higher succinics. And in general, if you look at this, if you look at the lows versus the average and the highs, you can see there's some big differences. For example, the sample that came in with gluconic acid at 9.65 grams per liter, obviously there was some microbial activity going on there. Likewise, the one with 1.31 grams of lactic acid, there was some microbial activity, and succinic. All right, so those are indicators. You can just look at the organic acid profile and already tell there's been some activity. And once again, in wines, and I should tell you that these are just raw data pulled off of our LIM system. So some of these are white wine, some of them are red, some have gone through ML, some haven't. So that's why the data, there's such a range. But still, if you look at these levels, these high levels, something with, gluconic, glucuronic, lactic, or succinic, that's all due to microbial activity. So once again, just by looking here at what's going on in the organic acid profile, you can see something's been going on. But it does change, once again, vintage to vintage, year to year, varietal to varietal. And chemistry is not the only thing that changes every year. What a lot of people maybe they don't think about, but microbial populations change dramatically. We did a study back in 2014 and 15 in the Willamette Valley where we were looking at native or non-inoculated fermentations. And it's a really interesting talk. So I don't know, maybe if people have an interest in looking at or learning a little bit about non-inoculated fermentations, it'd be a fun seminar. But what we did is we had 6 vineyards, 6 wineries, and they took the grapes from the different vineyards and processed them in the different wineries. And so we got clusters from the vineyards. The very first year in 2014, one of the vineyards forgot to send the clusters. But anyways, so out of 5 out of the 6 vineyard clusters, we identified 29 strains of Saccharomyces, okay, in 2014, 29 different strains of Saccharomyces cerevisiae that did not match any in our library of commercial strains. In 2015, we identified 32 strains that didn't match anything in our commercial library, which is up over 200 strains. When we compared them, there was only 1 strain observed in both vintages. So it's a very dynamic, what's in the vineyard is very dynamic as far as Saccharomyces, and even in non-Saccharomyces, particularly we're looking at Pichia and Hansennia spora, big differences in the same vineyard from one vintage to the next. So microbes change. Here's just a quick look. Lactobacillus conchii is a bacteria that comes in on the grapes. It's now been renamed to Apilactobacillus because the api from Latin for bee, because it's associated with beehives. But in 2022, we had some really big, 2 massive heat spikes in the beginning of September. And usually when it's really hot weather, we see more Lactobacillus conchii, probably because it's vectored by bees. We saw 50 samples that had greater than 10 million cells per mil of Lactobacillus conchii when they arrived for us to analyze them. So the juice, the grapes were crushed, they were put into a tank, they pull a sample, and in those cases, they actually completed malolactic fermentation before primary fermentation even started. So this is a phenomenon that we see once again in warm years. The point being that juice chemistry and juice microbiology are every year, they can be different. And so you're working with moving targets, especially as you're trying to understand acidity and acidity adjustments. So great fun. So anyways, I won't belabor this. I think Misha did a really great job of talking about this, but pH and TA are critical in winemaking, okay? TA, of course, is what you feel, that your perception of acid in the mouth. So it's a very important component of winemaking. And then pH is involved with everything, okay? If you've got high pHs, you're not going to be able to achieve the molecular SO2 you probably want, so you can't inhibit bacterial growth. There are also ranges that, below 3.2, most bacteria won't grow, although that's not so much of an issue, at least in California wines. It affects the solubility and stability of tartrate salts, okay? At higher pHs, calcium tartrate's much less stable. And so something to keep in mind. The effectiveness of SO2, as I mentioned, with bacterial growth, okay? You need molecular SO2, and as the pH goes up, the percent of molecular SO2 drops dramatically. Protein solubility, bentonite efficiency. Proteins in wine, if the wine has a higher pH, there's a parameter of proteins that's called the isoelectric focus point. And so when the isoelectric focus point, which is a pH value for the protein, is the same as the solution it's in, it is not soluble and it drops out. And it also, the closer your isoelectric focus point is to the wine matrix, the less net charge it has. So as the pH goes up, so a wine at pH 4.0, those proteins are going to have a lower net positive charge than they would at 3.5, so that can impact bentonite efficacy. Certainly, it impacts polymerization of color pigments, so your anthocyanins, et cetera. Oxidative and browning reactions are pH driven. And then wine aging reactions, okay? So tend to occur slower at lower pHs and faster at higher pHs. So all of these things, which are so important to wine, are influenced by pH. And so in the past, or if you did standard chemical acidulation, you would use tartaric or malic most likely. Commercial malic is half D, half L. So you put in a gram of commercial malic and malolactic bacteria consume half of it. But you get half a gram of stable malic. So people use that as well as tartaric. As far as deacidification, there's all kinds of ways: potassium carbonate, potassium bicarbonate, calcium carbonate, or the double salt. One that I recommend all the time is just cold stabilization. If you've got a red wine and your TA is a little bit high, just cold stabilize it some. In Minnesota, you just open the doors for a week in the winter. That probably take care of it. Out here, they do have to chill them, but that's another way of dropping acid. But the purpose of today's talk was biological. So how do microbes influence juice in wine? And essentially, deacidification by degrading existing acids, and then acidulation by producing acids from other juice wine components, and it's primarily from sugar, from hexose sugars. We'll talk about that. What I'm going to go on a little bit is just discuss the individual acids and that we're trying to increase versus the ones where we're decreasing. Okay, so lactic, acetic, succinic, and gluconic are increased by biological activity, and then malic, citric are decreased by biological activity. Acetic acid, I'll talk a little bit about that. That can be decreased by some biological activity. And tartaric acid, although tartaric acid is relatively stable, botrytis cinerea, and I'll mention that can consume tartaric. There's lots of ways for the acid to change. So when we talk about yeast lactic acid acidulation, and this was already mentioned a little bit, the only commercial strain that I know right now, or species that you can go out and buy today to acidulate, is Lachantia thermotolerans. And this can produce elevated levels of lactic acid from glucose, and they say 2 to 10 grams per liter, and that lowers the pH and increases titratable acidity. But Lachantia thermotolerans has a moderate, they call fermentative power, and it can't ferment in ethanol concentrations generally higher than 9 to 10%. So what that means is you generally requires a co-fermentation with a strain of Saccharomyces cerevisiae to get it to finish. It's difficult to predict the degree of acidulation, and this is important because I know people who used some of the commercial strains when they first came out, and they ended up, with Central Coast Cabernets with a pH of 3.2 and a TA of 12, where the wine where they didn't use the Lachantia, it was about 3.8, and I think the TAs were about a little under 6. So it's hard to predict the degree of acidulation, but they certainly have the ability to acidulate. There are several commercial strains available right now from Scott Laboratories, Lafort, and Guzmer. So you certainly have your choice, and they're either as single strains or in mixtures with other yeast. So once again, this is a product that's currently available, and you can go out and buy it, use it tomorrow. Bacteria, lactic acid acidulation. This is something where, Enococcus can produce lactic acid from hexose sugars. We generally think of Enococcus as something that's deacidulating. It's converting malic to lactic, so that's sort of a deacidulation. But certainly, if there's hexose sugars, they can produce quite a bit of lactic acid, especially if the pH is lower. They tend to operate in a homofermentive pathway where they produce 2 lactic from 1 sugar. If the pH is higher, and I'll talk a little bit more about that, they create a lactic, an acetic, and a CO2. Lactobacillus plantarum, which all of us know as Lactobacillus plantarum, but they once again changed the names, can produce lactic acid from hexose sugars. When they first came out with the commercial strains of Lactobacillus plantarum, Lactoplantobacillus plantarum, everyone was excited because there was no risk of VA fermentation. So they were doing, VA production, they were doing co-fermentations. But if the malic is all gone and there's sugar, they start making lactic. So they can actually, once again, overshoot your targets with these. So it's slightly easier to predict the degree of acidulation based on substrate level, but it's difficult to control. Now, what about acetic acid? I mean, whether you like it or not, acetic acid is a big component in the winemaking process. I know many winemakers talk about how it lifts the fruit, okay? I just think those are winemakers who their method or process ends up with higher levels of VA, and so they just accept it and then say they intended on having it. But in my observations over 30 years in this industry, I would say excessive acetic acid is the #1 microbial spoilage problem in the industry. And we have, I think, 5 companies in this area with many employees that make a really good living removing VA from wine. So I think I'm pretty spot on when I say it's the most prevalent problem. But it occurs throughout the process. So yeast species, so Pichia, Candida, Hansenia spore, they can all produce acetic acid, generally early on in the early fermentation stages. But what I would say is that if you get elevated VA at the very beginning of fermentation, some of that can be ameliorated by Saccharomyces cerevisiae, which has the ability to actually metabolize acetic acid at lower levels early on in the fermentation before there's any ethanol stress. Now, Pichia species, we see those having a problem, often producing elevated VA, and it's usually up to about 1 to 1.2 grams per liter. And it's right around that mid-ferment, 6 to 7% alcohol, because at that point, the Pichia is not capable of dealing with that alcohol stress. And so they start shutting down. It's usually rate limiting as the alcohol increases, and the faster the alcohol increases, the less acetic acid produced. Unfortunately, acetic acid produced after 6 or 7% alcohol is generally not metabolized by Saccharomyces, at least in what we've seen with these ferments where the Pichia has gone bad. Of course, we obviously can get acetic acid production by bacteria. The heterofermentive lactic acid bacteria like Lactobacillus brevis hilgardi, fermentum, Lactobacillus cunkii, and Enococcus can produce approximately 0.2 of a gram per liter of acetic acid from 1 gram per liter of hexose sugar. As a matter of fact, the biggest bump in acetic acid production that we see in a if you will, proper, normally done wine making process is at the end of malolactic fermentation. Because as a rule, most people feel if they're down below 1 to 2 grams per liter of sugar, the wine is dry, they can start ML. And then once the bacteria finishes up the malic and the citric and turns on the turns on to the sugar, you can make from 2 grams of sugar, you can make, and it's usually fructose at that point, you can make 0.4 grams per liter of acetic acid and causes a big bump. So that's something that's well documented. Unfortunately, if you have more sugar or if you've got a stuck or sluggish ferment, it can be a real problem. Acetic acid bacteria can produce quite a bit of acetic acid, but they need dissolved oxygen. It's got to be greater than 3.5 to 4 milligrams per liter. And usually the only place you see that is in perhaps the top 5 millimeters of wine in a barrel. And if you're doing a good job with headspace management and keeping your molecular SO2 up, then that generally isn't a problem. On the other hand, if you abuse the barrel and you've got 6 inches of ullage in the barrel and very low SO2, you can make a lot of acetic acid that way. So what about biological acidulation with others acids? So yeast produce a lot of different acids. So Saccharomyces cerevisiae can produce succinic, what we see primarily succinic and pyruvic, fumaric, some ketoglutaric, but succinic is really the big one, where we see 2 to 3 grams per liter of acid. There was a study published where they looked at organic acid production on a number of commercial strains and found that Vin13 produced the highest amount of succinic, and the rest showed lower succinic acid. But that was one study in one wine. And I can tell you, just from looking at client data and talking with clients, that we routinely see succinic acid go up by at least 1 gram per liter. And I don't know that there's that much of a difference in strains. There may be, but I just I see the end result where we see elevated levels of succinic acid. And then bacteria can produce other acids than acetic, and gluconic's the one that we see the most. And gluconobacter, based on its name, it does produce gluconic acid. So that's prior to the beginning of fermentation in the juice. So during cold soaks, or even if you've got some problems in the vineyard where you've got some sour rot or things like that, you can get production of gluconic acid. There's a new bacteria I'm going to talk about called Tatamella, which we just presented a poster at the ASEV on in Portland last week. We've done some work on that, and that can also produce elevated levels of gluconic. So we'll talk about that in a minute. So lots of places to get acid produced. You start out with a certain TA and pH and think you're all set, and then things can change fairly dramatically. Generally speaking, the biggest issues are too much lactic acid by Lachantia or Lactoplantobacillus, or elevated levels of acetic acid bacteria from the lactic acid bacteria. Now, into biological deacidification, this is, I think the one we're all most familiar with is using, bacteria to degrade malic acid, right? That's the biggest one. We do that all the time. But the same bacteria that degrade malic also degrade citric. And then, interestingly, Saccharomyces cerevisiae can consume acetic when there's no or low alcohol. And we've seen quite a bit of that. And as I mentioned earlier, tartaric acid, botrytis is very, very happy to consume tartaric. So if you're making a botrytis-sized wine, you may find that your overall pH is up and your TA is lower, and you have very little tartaric acid. So that's something that you need to watch out for. And as a side note, if you're doing any kind of lab chemistry and you have a tartaric acid solution sitting in your lab, be careful because it can get infected by botrytis. And if you notice your tartaric acid standard keeps getting lower and lower, it's probably because there's botrytis consuming it. All right, malic acid degradation, that's the big one, okay? So the primary route of malic acid degradation by lactic acid bacteria is an enzymatic decarboxylation. And that's the one that we all talk about with Enococcus eni or Lactobacillus plantarum or any of the lactic acid bacteria. And that's what we've been leveraging for years as a way to do biological deacidification. There's another bacteria called Tatamella, and I don't even know how to pronounce that. I'd say Tysios. I'd pretend it's like pterodactyl with a P in front that you don't pronounce. But this is something we've been working on here at ETS as a side project, and it's actually pretty interesting, and I'll show a couple slides on that. And then yeast can consume malic acid by converting it into ethanol through malloethanolic deacidification, and this is something that was Misha touched on very well, talked about it. And I don't know of any commercial strains of Schizosaccharomyces or Isotentica, but I think they are available. For research purposes, and there's been a lot of work done, a lot of publications on these, but I don't believe that Lallemand or Guzmer or Lafort have commercial strains on these at this time. That may be something coming out, but we certainly see these in juices quite often. Okay. So the malic acid degradation that we're most familiar with is Enococcus eni. It's a heterofermentive fructophilic lactic acid bacteria. That's how it's defined in Bergey's Manual of Bacteria. It loves fructose, and what's usually the primary sugar N, that's at the end of fermentation is fructose, so it's very happy with that. It's really well suited for wine conditions of pH and alcohol. Most strains are fairly tolerant up to about 15% alcohol. Certainly if you've got a very low pH white wine, it might be difficult because they tend to struggle below 3.2, although there are some strains that can go lower. But certainly, at least out in California with the red wines, they love the pHs that we have out here. They're intolerant of total SO2 greater than 50. And so if you've got something with a higher alcohol and a higher total SO2, and a lot of times people have difficulty getting through ML, and they say, well, I didn't add any SO2, but Saccharomyces cerevisiae can produce SO2 that gets bound up to the acetaldehyde also being produced, and you can get elevated levels of total SO2 even if you don't put any in. I think Montrachet is a strain of commercial strain which is well known for producing at least 50 milligrams per liter of SO2. So intolerant to that. But they're capable of metabolizing hexose sugar into lactic acid and acetic acid and CO2, and that's when you have a problem. And that's generally when you're over pH 3.6 or so, 3.7. So they can be risky to use for co-inoculated malolactic fermentation because if you finish ML before you finish primary, then you've got sugar and a big hungry population of Enococcus. It is currently the only bacteria authorized by the TTB for biological deacidification. And so technically, if you're going to do, malolactic fermentation, you can only use Enococcus. But that's changing. Okay, so Lactobacillus plantarum is formerly known as Lactobacillus plantarum, is the new kid on the block. And it's a homofermentive lactic acid bacteria, which means that it does not have the capability of producing VA. So that's a very attractive, parameter when you're looking at doing malolactic fermentation if you finish first with the ML. It's well suited for wine conditions of pH and alcohol. It metabolizes these hexose sugars, like I said, forms lactic acid, does not form acetic. It's ideal for co-inoculated malolactic fermentation, and that's what they're using it for. It's commercially available outside of the United States. I won't mention names, but I know a number of winemakers in the US who go up to Canada in the summer and purchase it, and I guess the term would be smuggle it back in, and then use it in their winemaking. It works really well. Now, I just got an email from a friend of mine who's a general manager at Scott Laboratories, and she said they just received GRAS status from the FDA last week. Okay, and the TTB has authorized the use of this commercial strain for experimental use under their Code of Federal Regulations 2724.249, which means experimental, which means you can't sell the wine. I don't even know if you're allowed to drink it. You're supposed to show how you destroy it, but at least that's the first step. And then they're hoping by next year to get 24250 status, which is sort of an approved status, so you can trial it and actually sell the wine. It's extraordinary how long it's been. I think I gave Lallemand a bunch of data probably 10 years ago on the prevalence of Lactobacillus plantarum in juice in wine in California, actually the United States, from all these samples. And yet it's taken 10 years just to get to this step. And by the way, it's cost Lallemand a lot of money, but they do have a strain that's commercially available, and hopefully in the United States it will be available in the next year or two. So what about this new kid on the block I was telling you about, Tatamella Ticeus, I don't know. Anyways, Tatamella is what we call it. It's a gram-negative, facultative, and anaerobic bacteria categorized in the Enterobacteriaceae family. Okay, it grows phenomenally fast in grape juice. I'll show you some of that data. Very exciting. It completes malic acid degradation in 48 to 72 hours. It's just amazing. It also tends to degrade lactic acid and produces tons of gluconic acid. So first it deacidulates and then it acidulates. Very crazy. I don't know if there's a use for it, but we do see it. However, it's very intolerant of SO2. So if you're doing low SO2 or organic or biodynamic winemaking, it could be a player for you. And it's somewhat tolerant of alcohol. So very cool. What happened was we noticed in 2022 that we were seeing some juices that had no malic as they came in, and they also had no lactic acid bacteria. So we could not explain the degradation of malic acid. So we, isolated some microbes and sequenced them and found these. And then in 2023 we saw the same event, and at this point we already had a scorpion probe for it, so we're able to identify this, and we isolated a few more strains. Oh, I should say it's also got temperature limitations, but the biggest thing is intolerant of SO2. So we set up some experiments just to see what's going on, and there's a lot of data here, but basically what I want you to look at is these are our different strain isolates, and this is what we inoculated at. So anywhere from 2,000 to 10,000 cells per mil. Within 24 hours, I don't know what happened there. Something went backwards. Okay, within 24 hours, it was over 10 million cells per mil in 24 hours. It just grew. I've never seen it grow so fast in my life. If you look over here, you can see our starting ML our malic was 4.51 grams per liter. After 24 hours, it dropped down by about half. Within 48 hours, it was mostly gone. And after 72, it was certainly below that 0.1 gram per liter. And there was no VA produced. If you look at we did a longer term study with it, looking at 9 days and 19 days post-inoculation, and you can see there was an initial TA drop and then an increase. The things to look at were essentially malic was degraded. We did see production of lactic, Now, at first, I was put off by this because I was thinking that we should have seen about, I don't know, 1.6 grams per liter of lactic produced for that degradation, that amount of malic. And there's like 2.6, was it 2.6 grams of malic? So we should have seen about 1.6 grams of lactic produced. But remember, the malic's completely degraded in 72 hours. And so in the next 4 or 5 days, it appears that lactic is being degraded. And in the day 19, the lactic is still dropping. So it's degrading malic, then slowly degrading lactic. And then the thing that we found very interesting was it produces massive amounts of gluconic acid. So all in all, a very interesting bacteria, and we were able to explain what was happening to client wines. So that was kind of fun. We got a publication out of it. And I don't know, Lallemande or someone might be interested in trying to develop a commercial strain of this for people who are, like I said, if you don't use SO2, it might be a very interesting way to complete malolactic fermentation very early before you even start primary. So there's a few other things that get degraded. As I said, commercial strains of Saccharomyces can metabolize acetic acid in the early stages of fermentation. In 2022, we had those high heat events. We had 2 days, it was about 5 days apart where the temperature was close to 110. So 2 consecutive heat events, and we had tremendous amount of sunburn. And we saw the most bizarre thing, along with those elevated levels of Lactobacillus conchii I showed you. We saw juice coming in, so grapes coming in being crushed, and they had extraordinary levels of acetic acid in the juice. And there were no microbes present that, could have caused that. We couldn't figure out what it was. And I'm talking 1 to 3 grams of acetic acid per liter in juice coming in direct from the vineyard. And so people were freaked out about it. And we just said, well, look, inoculate with a heavy duty commercial strain, it'll metabolize that. And that's what happened. They did. And every one of those, we watched the acetic go down. And then, they ended up with fairly normal acetic acids because once again, Saccharomyces can handle it early on. I think once you get past 6 or 7% alcohol, it doesn't deal well. And actually, as you get to higher alcohols, elevated acetic acid can stick a ferment. Then citric acid, lactic acid bacteria metabolize that. Like Misha mentioned, you can form diacetyl and other products. And then botrytis can metabolize tartaric. So, if you've got a lot of botryticized grapes, you crush everything open. Now you've got botrytis in there. You release tartaric. You can see a significant drop. Certainly, if you're doing any type of a cold soak or a slower start on your fermentation. So what I would say in wrapping up is be careful what you ask for when you're talking about biological acidification. There's a whole bunch of commercially available non-Saccharomyces yeast that can do a lot of different things, like prevent other microbes from growing, or having different glycosidase enzymes like the Mechnicauyas or Torulaspora, Lachantia for certainly making lactic acid, and some Pichia strains. So all these different commercially available non-Saccharomyces strains were developed to influence and change the wine. So that's interesting. But what I will tell you is that these are living organisms. They're individual strains. Each wine matrix is different, so it's very difficult to predict the outcome. Okay. In many cases, you'll still, likely have to use physical or chemical means of acidulation or deacidification to fine-tune your pH or TA if that was what you were using these for. A lot of people use them for, trying to release, more aroma compounds. And what I would say is that the sensory impacts, at least in my interactions with winemakers and from what I hear, is that the sensory impacts from the non-Saccharomyces yeast are more noticeable in non-oaked white wines as compared to more heavily oaked red wines. And that makes sense. Heavily oaked red wines, you're masking things. There's longer time in the barrel and everything. But certainly, some people are pretty excited about what they're seeing with these, non-oaked or lightly oaked, neutral oaked white wines with these different strains for aroma compounds. And then certainly, there's a lot of excitement, certain in California, where we generally have high pH, low acid wines for the Lachantia to be used. So you can do single, mixed, or sequential fermentation. Some of the commercial yeast providers already make blends for you, so that's kind of interesting. And so what I would say is this is certainly a tool you can use, and so have fun. And by all means, this is the fun part of winemaking. You've got to make a living, but you can always experiment a little bit on the side. Okay, but that's it. So thank you very much.
Well, thank you, Rich, for this great presentation. I just want to remind our viewers, if you have any questions, now is the time to please type them in the chat box, and we will answer them as we are able. But now we're going to go on to, in lieu of any questions, into our panel discussion. Do we have any questions in the chat box?
No, we don't. But thank you very much, Misha and Rich, for those great presentations. I think it was a lot of information, but it's bringing some questions. So please take time just to ask any question in the chat box. So now we are being joined with John Taylor and Kent Foulker to talk more a bit about what we do, what you do, what you do in your winery in terms of microorganism. What microorganism do you use? Have you used those different bacteria to deacidify mainly, maybe, or acidified? I'm not sure you're trying to acidify your wines, but what do you do currently?
Currently, I think there's multiple types of winemakers, right? There's some that are chemistry forward, and there are some that are chef forward. And after watching this beautiful presentation from Rich and Misha, it was I'm definitely a chef style winemaker. So I do a lot from Scott Laboratories using Lalaman products and things along those lines. I co-inoc with malolactic fermentation with a lot of our reds here. Our winery does a lot of hybrids. We also do a lot of vinifera, bringing in whole cluster grapes from the West Coast. So we do a little bit of everything. Using any experimental products? No, not at the moment. We're, like I said, pretty stuck to that Scott Labs book that helps us out a lot in staying safe. Like I said, the in-depth presentations here definitely lets me know that I have a lot more to learn when it comes to the chemistry and the different types of acids and all their basic chemistry forms. So like I said, we stick pretty hard to that book, and that helps us out and keeps us in the safe zone most of the time.
Yeah, I've being a consultant, I get to see a lot of different methods in terms of how acid is being managed, both from a chemical as well as a microbiological standpoint. My personal belief is I have not used a lot of co-fermentation or yeast strains necessarily that help deacidify, either by taking malic out or changing the matrix to some degree. I have focused primarily on in hybrids, in cold climate varieties. I have focused on aromatic potential and basic chemistry in terms of looking at alcohols and how that balances with pH. Having clear targets in mind in terms of what types of styles of wines are being made, and then choosing appropriate microbiologic agents to achieve those through. I will occasionally do malolactic inoculation under 5 Brix in fermentations, so 5% sugar, but I have not had nearly the success with co-fermentation in achieving the complexities, particularly in red wines.
Right.
I'll just kind of jump in on that too, John. Obviously we here in Iowa, we also work with a lot of hybrids, like Marquette and Frontenac and Brianna, Edelweiss, that whatnot. We are kind of, you have a style in mind that you want to work with, and then you got to work backwards. You have a style that you want to finish, you have a plot of land, and you have your varieties on the ground. And then it's work backwards. Are you going to barrel age? Are you going to go through malolactic fermentation? Are you going to have residual sugar? Are you going to have, are you going to batonnage? There's lots of different things that you're going to do going backwards that you need to look for a certain pH and a certain TA as your base must or juice. And so choosing on when you're going to harvest, are you going to blend? Is it going to be a single varietal? All of those things, you got to work backwards in order to make the right decisions on what bacteria strain you're going to use, and what type of yeast strain you're going to use. Are you going to acidify, deacidify, things along those lines? You got to work backwards when you make that. And that's kind of what we do here too.
Yeah, that's a great point, Kent. I think, again, having that style in mind and then working backwards, like you said, to get to that point is huge. I would just add to that, sometimes you have styles in mind from a particular lot of grapes, and as we all know, Mother Nature is going to have her say, and the grape showing up at the door or the harvest decision may throw that plan in a couple of curveballs. So making sure that you have options other than what your intentions are is a huge part of this.
Thank you both for sharing your experience. We do have a question in the chat box. So Jason is asking, if we want to use some of the strains that are intolerant to SO2, what else can we use?
Are you talking about when I mentioned the Tatamella, which is intolerant?
I think so.
Yeah, well, so it's interesting. Tatamella, there were two really interesting papers on the grape microbiome published where they said Tatamella is the most prevalent bacteria found on grapes. The bacteria no one's ever heard of. Okay, it's the most prevalent. And they mentioned seeing alterations in the chemistry, the juice chemistry, but they never drilled down to find out what it was. So when we started digging into this, we saw, well, it's the most prevalent bacteria that no one's ever heard of. So it's probably on your grapes. And so, but it's intolerant of SO2. Even 12 milligrams per liter of SO2 at the crusher will knock it out. But we found that we could re-inoculate that one after a week and it would grow, whereas everything else, 25, 35, 50 milligrams per liter, it just didn't grow and wouldn't grow later. So it's intolerant of that bound SO2. But I would say for people who would like to, experiment with something like that, well, first off, if you just ran, and a small batch, just, don't add any SO2 into it. We do have an assay for Tatamella, so we could certainly check and see if it was there. But I guess the thing would just, if you bring it in and there's no malic, the malic's degrading, you still have to wonder, is it the Tatamella or are there lactic acid bacteria? So I think these things will occur in a natural fermentation with no SO2. I think the Tatamella would handle up to about 5% alcohol. So it'll be there and that population will explode, grow very quickly. But if you aren't actually monitoring for the presence of that microbe and the absence of other microbes that could cause the same thing, and you're not looking at the chemistry, you might not actually ever know it was there. But certainly that is something that we're going to be looking a lot more into this year.
That's great. Thank you. And it leads to one question that I have about is there any maximum of malic acid concentration where the bacteria are not able to be efficient? I'm just wondering if we have too much malic acid in the juice, even if we do co-inoculation to help a little bit starting with the bacteria, is there a max where the bacteria, you can do whatever you want, is not going to be efficient?
Yeah, I've been in the wine industry looking at data for a long time, and maybe not so much in my previous research position, but at ETS now for 18 years, I've never seen a juice or a wine that did not finish ML because of the concentration of malic. Oops, sorry. I have seen, there are many cases where malic won't start. Okay, that happens all the time. But usually once you get it going, it finishes. The only thing that I could say is if there was a condition where, and I don't know if this exists, but if there was some type of repression from lactic acid, because if the lactic acid built up to a certain level and maybe there was some kind of feedback mechanism, but I haven't ever seen that occur.
I think if you have too much malic acid, I mean, I think your pH probably would have been way off anyway. Your actual must would have been probably too acidic anyway for the bacteria to even start.
Yeah, 3.2 is about the line where I don't think any of the commercial strains will go below 3.2 right now. There may be a new one out there that I'm not aware of, but most of the time that was always the line that we drew. So you're right. If you had a lot of malic, then you probably, if the pH is too low, it could be very difficult.
Yeah. Well, Ode, I would say I definitely saw Norton's peter out more than any other cultivar that I've ever experienced. That was a more common thing that we were doing some outreach testing, and if there was one that didn't make it through, it was probably Norton. I can't say that it was purely just the amount of malic was there. It could be a combination of missing some other nutrients that it needed to be able to keep going for that extent, or some other factor. But I do think that there are some weird things that happen with some of the non-traditional cultivars that you always keep you on your toes.
Yeah, absolutely. And once again, I have no experience. I don't know of anyone in California growing Norton. So, but I do know that there's definitely big differences between some of these varietals as compared to traditional Cabernet, whatever, as far as acidity. I think, isn't Norton a fairly acidic wine? High TA, lower pH?
No, high TA and high pH.
Okay. Oh, that's an odd one. Okay, good.
It can be quite challenging.
Glad that's your problem and not mine.
Yeah.
Yeah, I honestly, I thought people were lying to me when I first came to Missouri, and they said, "Oh, so it comes in at pH 4 and with a TA of 13." I'm like, "You're doing something wrong." And then it was like, "Nope, nope."
Yeah, I'm glad I don't have to deal with Norton up here in the frozen tundra. We just have the classic, the Frontenac family of grapes. They can easily get up to 26, 27 bricks, but still be at 3.1 and up there at 9, 10, 11 grams per liter of acid. And I'm not shy to say it. Water can go a long way to bring that bricks into a more balanced range, to possibly have a slight effect on raising the pH and certainly lowering the acidity a bit. I tell a lot of the winemakers we have here in the Midwest are newer or not traditionally educated, and the first time you bring up amelioration or at Gallo, we used to call it a WAF, water-aided fermentation, post-harvest irrigation. It's quite common, and I've added in California. I added quite a bit of water at different places. But again, a newer winemaker who's very idealistic and very purist, they tend to balk at that until you sort of have to tell them 2 or 3 times. And then they do it and find out that there's one thing that Frontenac grapes never lack. They've got plenty of color, plenty of flavor, plenty of body, and so a little water can go a long way. But that all said, my mind is now spinning with and you say that is going to become TTB approved. Oh, you're muted, Rich.
Yeah, Nicola Hall, just because I've been working with him for a long time on this, and she just sent me an email on Friday saying they'd achieved GRAS status from the FDA. So that's the first step. And then TTB is granted them that 249 status so it can be used. So, but if you think about it, that means as a winemaker, you've got to fill out all this paperwork.
Mm.
You've got to monitor where the wine goes, how you dispose of it, all of this stuff. It's really, but that's what it is. So like I said, there are a number of people who go to Canada and bring it back in and use it and with great success. So
I would.
so it'll probably be in 25 or 26 when we'll be able to buy it here in the US and use it.
I'm regularly in touch with Nicola, and
Yeah.
and so I'm sure I'll hear about that at some point. Yeah.
We have a question in the chat box. So what are your thoughts on cold stabilization of white grape juice immediately after pressing it and inoculating it with a good strain of Saccharomyces to decrease tartaric acid?
I don't know that the only way you're going to decrease tartaric acid with the fermentation is because as the alcohol goes up, the potassium bitartrate is less soluble. But I mean, that I don't know of any way, if you, Saccharomyces, to the best of my knowledge, and I never say never when we are talking about microbiology, but to the best of my knowledge, I don't know any Saccharomyces strains that degrade tartaric. Okay, if that was the question. But certainly, as in every wine, as you increase the ethanol concentration, you're going to, start dropping out potassium bitartrate. So you do lower the tartaric that way. Sometimes people are quite surprised at how low the tartaric is in a finished wine as compared to the start, but that's simply because of that precipitation of potassium bitartrate.
That was a revelation when I came to the Midwest from California. The first person who said to me, "Are you going to cold stabilize your Marquette?" And I sort of laughed and said, "Oh, of course we never cold stabilize red wines." And this person who knew a lot more than me, which is not unusual, reminded me that, well, in fact, you can cold stabilize a red wine, and it will have the same effect. It will precipitate out some excess tartaric. So that's been very useful over the years. Not everybody does it. You can lose a little bit of color, I've noticed from time to time doing that.
Sure. Yeah, we see that, but we do. We have a lot of clients who just want to drop the TA on a red wine a little bit, and they'll use cold stabilization simply because versus adding potassium carbonate or bicarbonate,
Yeah.
it's a pretty rough touch on the wine.
Yeah.
So they'll just cold stabilize it, and you can drop TA a couple tenths pretty easily. So
Yeah.
It's a good method too, and you do that for a rosé. I did a petite pearl rosé and cold stabilized it, and it dropped a lot of color and made it actually a very pretty.
Okay, yeah.
rosé from it.
Yeah,
Potassium
timing of
bitartrate
cold
binds up
stabilization
color.
is always interesting because what a lot of people don't realize is that if they're in a traditional barrel aging environment, you will be getting some tartrate crystallization in the barrel, and it's not a purely scientific method because you won't be completely cold stable coming out of barrel, but it will change your tartrates to a degree coming out of that. So that's something that's important for people to realize, is that what goes in is necessarily the same thing that comes out of barrel in a colder barrel storage environment. The other thing that's fascinating is pre cold stabilizing by freezing juice and bringing it back, thawing it out, and then fermenting after harvest. That also is a method with which you can cold stabilize prior to fermentation, and it does change the environment quite a bit.
Are you talking specifically for reds or whites?
Whites and rosés typically.
Okay.
Yeah. So if you're looking for a different matrix to start out of the gate, that's one method. It does add cost, of course, because you're freezing juice in smaller movable totes, of course, but it is a method with which to manipulate that environment and get to still have fermentable controlled fermentable sugars on the backside of cold stabilization. So because I've seen native yeast kick off at 32 degrees when held for longer than 3 weeks. An active fermentation at 32 degrees. So it's one where you need to be very careful if that's the method that you're choosing to use. But I've found that freezing the juice is, if you're wanting to cold stabilize prior to fermentation, that's the best method to achieve that.
Very innovative.
Costs you about $25 a pallet footprint. So if you get a 330 gallon and fill it with 300 gallons worth of juice, and you get about 1,000 gallons worth of juice for about $75 for the month. So economically, it's not too terrible. You're talking about about 15 cents a month in fees there per gallon.
That's not an aspect I think most people normally consider to have such fine tuning on the cost of production and cost of cold stabilization. That's some useful calculus.
Right.
Uh,
Thank you very
Rich,
much.
Rich, we're going to have to get you out here sometime to enjoy a nice La Crescent wine or a barrel aged
Love to.
Marquette.
I still have friends back in Ames, so I would love to. I thought we were going to get a chance to come back this summer, but it fell through. But I'd love to come back sometime and pop up and see you as well. Although you know that if you're in Iowa, you make jokes about people from Minnesota and vice versa. So used to be at the Minneapolis airport, they had the book jokes on one side. It said jokes about Iowans. The other side it said jokes about Minnesotans. Yeah. So anyways, but yeah, I'd love to come back sometime.
Yeah.
It took me about 6 months, Rich, to realize that the border battles in the Midwest are a real thing. I'm also originally from California. And I'm like, I don't understand this. And then I'm like, why is Wisconsin not like Minnesota? And Minnesota's not like Iowa. And no one likes the Dakotas. I don't get this.
Yeah, I know. But it gives them something to do in those long, cold winters.
Bragging rights. It's all about bragging rights.
Yeah. All right, well, thank you very much, everyone. I have to catch a plane, so thank you, and please, look forward to doing this again. Okay.
Thank you, Rich.
All right.
Thank you very much, Rich, for
Safe
your
travels.
great presentation.
Okay. Bye-bye.
That's time to conclude. So thanks very much to both our presenter, Rich and Misha, for your great presentation. It was a lot of information, but very useful, especially for the season that is coming. So thanks everyone for participating, and we hope you develop new knowledge. So now please send us an email if we didn't respond to all of your questions, or if you just have a question that comes up. Stay tuned for the next webinar. We're going to talk about the alternative products next time. So next month, July 23rd, we're going to talk about verjus and vinegar. So we are staying on the acidity topic, but talking about 2 other products. And then in October, we're going to come back after or closer to mid-season, mid-winemaking season. So we're going to talk about low alcohol wines and in October, and then challenges of color management in wines in November. And then December, we still have to decide what topic we want to talk about. So if you have any suggestions for topics, please email us or just complete the survey that you can have access to if you scan the QR code that you have here. Otherwise, I will send you an email, all of you, the link to the survey. So that way you can complete that questionnaire. So please just take 2 to 5 minutes max to complete the survey. And so, yeah, that's it for today. So thanks again, everyone. Thanks, Kent and John, for your great input and experience sharing. And we hope you can join in the future next month.
I agree.
Thanks, everyone.
Thank
And
you
thanks,
all
Drew.
very much. We'll see you next month, I hope.
Yes.
Keep in
everybody.
mind that verjuice and vinegar doesn't take much time. You can go from grape to saleable product in a very quick turnaround. So people are looking more and more at these alternative products. So hope you will join us July 23rd. Adios.
Thank you. Bye.